All right, it is now recording. And again, we're going to go over DNA replication today. You have gone over this in lecture, so hopefully you know quite a bit. The information in the packet, there's a packet that was placed on Blackboard, will be on the final test, okay? And remember, next Saturday we don't have class because it is Thanksgiving holiday. But this is all about DNA replication. So before we get started, I know you've heard about DNA, right? And what is DNA? That's the main stuff that's in your nucleus that is makes the chromosomes up. So remember, we've already talked about chromotin and chromosomes, which are the same thing, only chromatin's disorganized and chromosomes are organized. They're made up of DNA, deoxyribonucleic acid, and some proteins called histones. We're going to talk about how DNA replicates. So remember when I was taking you through mitosis and we talked about S phase, interphase? When you duplicate DNA replication occurs there. So it's all about DNA, how it replicates. So let's get into the packet. Okay. All right. Does everybody see the screen?
Yep.
Does DNA replication? Yes?
Yep.
Okay. So here we're going to go through this packet. This is the packet we're filling out. So I hope that you have it. Because this is going to be completed today, and there's several pages of this. And like I said, I'm going to talk about it tonight. I don't know if you printed it out or not, but it needs to be completed. So you'll have to complete that. And then I'll put on a completed one sometime this week. I'll get it on there. And I'll get it on before the weekend, or maybe on the weekend. That's what I'll do. I'll have it on before next Sunday, because you don't have class on Saturday. And then you can make sure that you have all the right information. But I want your one completed. So bring your completed packet if you can. Does everybody have access to printing information out?
Yep.
Okay, then you need to do that. This information will be on the final. All right, so let's get started. It says DNA replication activity guide. So we were basically going to use some models, but we don't have those here, but that's going to still be okay, because as we go through it, you'll understand. And then I'm at the end of this packet, we're going to go through DNA replication again, which is another exercise that you'll be doing on the actual final. All right. So let me just get acquainted over here. So it's all about deoxyribonucleic acid and replication in this one. But before we do that, we have to really talk about the structure of DNA and some information. So with what you've learned in lecture, you know that your cells are replicating inside of you by mitosis. Now, why are your cells replicating inside of you? So one cell will divide into two exact ones. We went through that whole entire scenario. And that includes, you will be duplicating the nucleus, right? So through mitosis, the whole thing, we have interphase, and then you have cell division, mitosis, and cytokinesis. Through that whole entire process, one cell becomes 2 identical ones. Now, why? It says number 1, it says list at least 3 reasons why a cell must undergo division. First one would be is if some cells were removed from you and it was unintended. So like through an injury, if you lost some cells, you want to bring those cells back. So your cells are going to divide to form exact cells that were like it. That's one reason. Another reason is if the cells got injured without removal, but they died and they shouldn't have. So if like you got a burn or some type of injury through the cell where they died for them to come back, even like a chemical that could kill them. So that's another reason. And the third reason is if you wanted to grow in size, as you grew wider and taller, you'd want to deposit like cells in areas from which they came. So in question number 2, it says, imagine you are cutting a bagel, one of the most common household injuries, and you get a cut. The cut heals. How do the new cells compare to the original pre-cut cells? It would be exactly the same. Unless the injury was hard enough that your body had to lay down scar tissue. But here we're not going to say that. We're going to say, because your body prefers to repair or replace cells with the exact same type of cells and material that was removed. So we're going to say that they should be identical. Let's say this is a small cut. How does your body ensure that the new cells are the same? Because you're going to be going through interkinesis, mitosis, and cytokinesis. So by going through that whole entire cycle, we're going to make sure that the cells are the same. We're going to start first, and the cell's going to say, okay, we divide. We're going to duplicate the DNA. And after we do that, we're going to do a nuclear division, which is mitosis, and then the remainder of the cell division called cytokinesis. So that's how it ensures those cells are going to be the same. How does the DNA get into the new cells? Oh, well, we saw that in interphase, right? We're going to duplicate the DNA, and then through the process of mitosis, we're going to divide the nucleus in 2, which has the DNA in it, and then you'll divide the cells in 2. So they're going to get into those new cells by that whole process as well. So that was kind of sort of like a pre-intro that we've done a lot of that stuff already, of course, that you know, and there's an assumption that you know this from lecture also. All right, so introduction. No molecular structure has gained more worldwide recognition than the DNA double helix. So you know that's the structure. A double helix looks like a twisted ladder. The famous Nature paper written by James Watson and Francis Crick in 1953, entitled Molecular Structure of Nucleic Acid, ends with, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Since then, many scientists have focused on researching the mechanism of DNA replication. So what does this say? Watson and Crick, and there were other people involved too, discovered the structure of DNA. They mapped it out. They won the Nobel Prize. And I know that you've learned about this already. And we're going to go through it again. But it has, DNA is made up of nucleotides. And each nucleotide has a sugar, a phosphate, and a base. And those are going to be following in the subsequent pages on here. And then we'll talk about pairing of the bases with one another. All right. So this is modeling DNA replication activity. It says in this lesson you will learn how to copy DNA and you will learn how a copy of DNA is replicated for each cell. You will model a 2D representation of DNA replication using the foam nucleotide pieces. Well, we would have done that, but we're going to do it virtually. Okay, so I want you to look at DNA is 2 strands. So you could see a template strand and a non-template strand here. Look at the template strand. It has a 3'. That little arrow with the C thing would have been what these nucleotides would have looked like. But you don't have that, but that's okay. But the template strand starts with 3' on the left and goes 5' all the way on the right. The strand below it is called the non-template strand. That starts with the 5' on the left and goes all the way to 3' on the right. Are you guys seeing this? Yes or no?
Yes.
Okay.
Yes.
All right.
Yes.
So now I'm going to blow this up a little bit to show you. Now, so each one of these little spaces is a nucleotide, okay? Now, they're only showing you the base of the nucleotide because that's the only thing that differs. So let's just talk about each nucleotide, and you'll see this on the page following. Each nucleotide contains the sugar deoxyribose, a phosphate group, and a base. Because the phosphate group is always the same and the sugar is always the same in the nucleotide, it's the base that changes. So when people frequently write DNA, they just write the bases, but they know that each one of these represents a nucleotide. Why do they only write the base? Because that's the only thing that's different on it. What they want you to do is take a look and see that these strands are anti-parallel. One goes 3', 5', the other goes 5', 3'. You're like, what does that mean? You should know that from lecture, but I'm going to go over it. Just hold on pages that follow. But because each one of these is a nucleotide and we only write the base here, we know that the nucleotide with the base C, base pairs, and will be across from a nucleotide that has G. C stands for cytosine. So this is a cytosine DNA nucleotide. So across from it would be a G nucleotide. So it would always be C with G. And wherever you have a T nucleotide, so here's a T, across from it would be an A. Are you familiar with this? Yes or no?
Yes.
Yes.
Yeah. Okay. So here they're talking about base pairing between nucleotides. Now, if I don't hear any no's, that means that everybody knows what I'm talking about. So if you don't know what I'm talking about, please say no now, because we're not going to be going over all of this again. And remember, the next time I see you is the last time I see you before the final. And we'll be on to a whole different topic. So make sure if you don't know this, don't just say nothing. Make sure that you respond, okay? All right, so these are two strands of DNA, and in real life they would be twisted like a twisted ladder. These are the bases, and these are like the rungs of the ladder that you would be stepping on. Those are where the bases are. So they're saying pair the nucleotides of the template strand to the non-template strand of DNA you constructed. So you would have put this one together already to create a double-stranded DNA molecule. So what do they really want you to do? They want you to put the nucleotides that are missing. So what are you going to put across from a C? What would this be?
Guanine.
So it would be a G, right? A guanine nucleotide. So all the C's would be base paired with G's, and all the G's would be base paired with C's, right? Cytosine nucleotides. All the A's would be base paired with thymine nucleotides, and all the thymine nucleotides would be base paired with adenine. So you need to fill this whole thing out, okay? Did you guys print this out? Yes or no?
Yes.
Yes.
Everybody did?
Yes.
You did? Okay, start filling it out. I'm going to give you a little bit of time to do this. So Fayez, did you print it out, Fayez?
Yeah.
Okay, and did you print it out, Champagne?
Yes, I did.
Okay, all right, so I'm assuming I'm just randomly picking people. All right, I want you to fill this out. So let's just do it, guys. So this would be a G, this would be a G, a C, right? A G, a T, an A C, an A C, an A C, a T, a C, a T, an A T, a G, a T, two more A's, two more C's, two more G's, two T's, a C, a T, a G, a T, a G, an A C, two A's, a T, a C, a G, an A, and a G. Now remember, these aren't just bases. We're just writing those in there. These are the nucleotides are named with respect to the base because that's the only thing that changes in DNA. DNA nucleotides, which form DNA, have three parts: a sugar, a phosphate, and a base. The sugar and the phosphates are always the same. They're phosphate and deoxyribose. That's a sugar. But it's the base that's different. So you have cytosine nucleotides, guanine nucleotides, thymine nucleotides, and adenine nucleotides. And they are symbolized as A, C, G, and T. Okay, so they want you to record the template strand. Yes?
Sorry, are those lines the nucleotides?
Yes. All
Okay.
these dots, you need to put the nucleotide in. And it's
Okay,
base
thank you.
paired to it. So this would be a guanine nucleotide, and it is base paired with a cytosine nucleotide. Okay?
Okay.
So they want you to record all of those in the blank spaces provided above, and you're making the template strands. You're just going across, just put those in. And then afterwards, examine the strands. What do you observe about the arrow ends of the molecule? Now, this is where, these aren't really there, but in our foam models, they would be there. Well, this arrow, wherever we see it, always points to the 3' end. Now, you don't know what that means, but the arrow right here, see that little arrow? You could tell that that would be the 3' end of the strand, okay? And what does the arrow indicate? The 3' end. We'll just leave it like that because we're going to get into the actual structure of the nucleotides. It's coming. So I think that's good enough right now rather than going back and forth. Are you guys all done filling in your nucleotides? Yes or no?
Yes.
Yes.
Okay.
Yes.
All right. So we're going to go on. Anybody need more time? All right, now let's look here. This is where it gets pretty cool because it really talks about the structure of the nucleotide. So remember, this whole thing is a nucleotide. This is a nucleotide. These are the things that come together to build DNA, which is a nucleic acid, the same way that amino acids come together to form peptides and proteins. So let's look at the nucleotide. I'm going to start here with this is the sugar, okay? And you're not going to draw this sugar. Don't worry about the sugar. But you do have to know that the sugar in the DNA nucleotides is deoxyribose. This deoxyribose is just a 5-carbon sugar. And what they do with the sugars is they label them as primes. So this is the one, there would be a carbon here. And this is the 1' carbon. Here's another carbon here. That's the 2' carbon. Here's another carbon here. And you're like, I don't see it. Of course you don't see it because in organic chemistry, this is standard way of how they draw organic molecules. when you have things that come to a point or intersect, there's a carbon there. So there would be a carbon here, a carbon here, a carbon here, a carbon here, and then they have to make the carbon out here because that's the end of the sugar and there's no intersection. So let's see how they numbered the carbon. Now this whole thing is deoxyribose. Here's the 1' carbon. Here's the 2' carbon. Here's the 3' carbon. Here's the 4' carbon. And here's the 5' carbon. Now I'm going to tell you what's attached to these because you need to know it. At the 1' carbon is where there's a nitrogenous base, or the base. And I'll take you through what those bases are. Adenine, thymine, guanine, or cytosine. And we're going to look at them in a minute. But this is where the base is. The base is always attached to the 1' carbon of deoxyribose. So remember, this whole thing is called what? A nucleotide. This whole thing. It has 3 parts. A sugar, called deoxyribose. A phosphate group, which is here. And a nitrogenous base, which is here. The nitrogenous base, which could be adenine, thymine, guanine, or cytosine, is attached always at the 1' carbon of deoxyribose. The 2' carbon has a hydrogen. The 3' carbon has an OH group. This is called a hydroxyl group, OH. 4' carbon is going to be attached to the 5' carbon and also a hydrogen. But this 5' carbon is attached to the phosphate group. For this class, all of the carbons are important, but the ones especially to know are the 1' carbon will have the base. The 3' carbon has the OH group or hydroxyl group, and the 5' carbon has the phosphate group. Are you staying with me? Any questions on that? Because this is going to be on the final. What's at the 1' carbon, the 3' carbon, and the 5' carbon in nucleotides. So,
what's on each one again?
what?
Can you repeat what's on each one again? I get that
You can see it. You tell me. What's at the 1' carbon? What do you see it attached to with my arrow?
The nitrogenous base.
Yep, the base. So what's the base? A, T, C, or G?
Mm-hmm.
All right, let's go to the 3' carbon. Now, it's attached to this hydrogen there, but the big thing is it's attached to an OH group. See it?
Yep.
Okay. And the 5' carbon is attached to the phosphate group. Okay. Now, I'm not going to be testing you anything about nucleoside monophosphate, diphosphate, triphosphate, but this just tells you here, there's only one phosphate group here, so it's called a nucleoside monophosphate. When it has 2 phosphate groups, it's a nucleoside diphosphate, and when it has 3, it's a nucleoside triphosphate. But then when you add the base to it, it becomes a total nucleotide. Okay? All 3 components. This whole thing is a nucleotide. Nucleotide. The big thing here is to know this is deoxyribose. The 1' is the base. 3' is the hydroxyl. 5', and these are the carbons in the sugar because the deoxyribose, see the O's? That's a sugar. 5' has the phosphate. Okay, now let's take a look at these different nucleotide bases. And they all have nitrogen in it, so they call it same nitrogenous bases. All right, here's adenine, and you do not have to know the structure of this. But this could be here. Or here's guanine. Now, these have double rings. See, there's 2 rings here. Guanine could be here. They look pretty similar. Where do they differ? Here they differ. And over here they differ. Now let's also look at cytosine. Here's cytosine, one ring, or thymine. Now, why do they have uracil here? Because that could be another base, but it's never in DNA. It could be an RNA. I wish they wouldn't have put that in here, but that's okay. With RNA, if we were talking about RNA, you would have a different sugar called ribose. Okay, so just for sake, because we'll be on RNA next week, this would be ribose instead of deoxyribose. Got the phosphate on it still, no big deal. But the nitrogenous bases for RNA could be adenine, guanine, cytosine, or uracil. But uracil is not here in DNA, thymine. These are the four that could be in DNA. Adenine, guanine, thymine, and cytosine. There's no uracil in DNA. Okay. Are we all right with this?
Yes.
All right. So now we're going to go on. So this would have been had you had your molecules with you. Okay. So we wouldn't have had molecules like this, but we would have had them like this. I told you, this was a nucleotide. But look at here. This is what, if we just compare, if you had these little different types of models, because many different come. This is the foam molecule, and this would have been like a plastic molecule. Tri-C doesn't have these. But this is the sugar part, okay? This gray stuff is the sugar over here. Right here. And then you can see some of the carbons are labeled. They didn't label the 1' carbon, though. The 1' carbon should be here because this whole thing is the nitrogenous base. And the nitrogenous base could be adenine, thymine, guanine, or cytosine. This is deoxyribose sugar. Here's the 3' carbon. Here's the hydroxyl group, OH. And here's the 5' carbon with the phosphate group. So they're saying the hydroxyl group, OH, shown in the above illustration, has been added to the photo of the nucleotide, since this particular model of the nucleotide does not show the hydroxyl group. I don't know. I guess, but it looks like it does. I don't know what this means. I'm sorry, because I see the hydroxyl group right here. It's strange. Okay, so again, where's the sugar in here? Here. Where's the base? Right here at the 1' carbon, right? Which would have been right there. Here's the 3' carbon in the sugar with the hydroxyl. Here's the 5' carbon in the sugar. Here's the phosphate group. Now let's take a look at our foam molecules to orient. This whole thing is the sugar, okay? So this would have been a 3' carbon. Here's the hydroxyl group, okay? Let's go back. And they've got this right here. Here's your sugar in the nucleotide, deoxyribose. This is the 1' carbon, 2', 3', 4', and 5'. This whole entire thing is this thing here. This is a nucleotide, this whole thing, okay? Now, they're just designating the phosphate group as, it looked longer before they kind of made it shortened. This is just the phosphate group, so this is the 5' carbon on deoxyribose. So here's the phosphate group, and again, remember, this is a whole nucleotide. This is a nucleotide too, okay? So this over here would have to be the 1' carbon, because here's the base, okay, right? If this is the 1' carbon with the base, A, T, C, or G, that would have to be here. This is the 3' carbon with the hydroxyl, so this would be right here, okay, the 3' carbon. And then the 5' carbon would have to be here because this is the phosphate group. Okay? Are you okay with that? Yes? Everybody see that?
Yes.
Okay. All
Yep.
right. So remember, this whole thing is the nucleotide. So this is, you got the sugar in here, right? You got the base in here, and you got the phosphate group in here. Okay. They said examine the diagram to the right. Circle and label the 3' carbon. Which one would you circle? It would be this one, right? This is the 3' carbon. And then they want you to circle and label the 5' carbon. So this is the 5' carbon. You should be doing this on your handout. Okay. And they say primers are used in numbering carbons on the sugar. I already told you deoxyribose portion of the nucleotide to distinguish them from the nitrogenous base carbons. I told you we only designated the carbons with primes for the sugar part. All right. So let's go down here. In the image to the right, label the locations of the 3' and the 5' carbon. So this is where the 5' carbon would be, and this would be the 3' carbon. How are the 3' and the 5' carbons oriented in the strands of DNA molecule you assembled? They're opposite to each other, aren't they? They're opposite. Did you guys finish this? Can we go on?
Yes.
Yep.
Okay. So remember, this is the 3' carbon because that's where the OH would be. This is the 5' carbon area because that's where the phosphate would be. And they should have put 1' in here, but they didn't because this is the base, okay, where the base would be. And on this diagram, here's the 1' carbon, 3, and the 5. All right. All right, now, when we're on this page, this shows you how the strands are oriented, and this is true. Okay, so we're going to start here. This is the first nucleotide, right here. Here's deoxyribose sugar, here's the phosphate group, and here's the base. This is the 1' carbon, the 3' carbon, and the 5' carbon right here. What base you want that to be? Let's say it's going to be A, right? Say it's A, adenine. I'm just going to write this because when we make the other one, they have to match. All right. Now I want to show you, this is the importance of the 5' phosphate, okay? So remember, the phosphate's always attached to the 5' carbon, and the 3' hydroxyl. This is the second nucleotide, right here. Here's the deoxyribose sugar. Here's the 1' carbon with the nitrogenous base. Here's the 3' carbon, which would have the hydroxyl. The H is missing because it's been attached to something else, but it was there. But just remember, the 3' had the OH group, and then the 5' had the phosphate group. Now I want to show you how these join. Pretend that this has an H on it. It's separate. It will form a bond. So this is the 3' hydroxyl. Absolutely. The 3' carbon hydroxyl, this would have been an H here. We talked about that previously. This is the 5' carbon with a phosphate group. These two things, this would have been an OH, and this is a phosphate group. They form a bond. 3', 5'. Where's the 3' carbon? Right here from the first nucleotide. Where's the 5' nucleotide from the second nucleotide? Right here. This is the 5' phosphate with the 3' hydroxyl. They form a phosphodiester bond. You don't have to know the name of that bond, but they join, and now they are linked together. Let's make this nucleotide G. So this was an adenine nucleotide, and let's make this one a G nucleotide. Are you with me? Yes or no?
Yeah.
Okay.
Yep.
So you understand that hydroxyl group, let me go back up here. The hydroxyl here at the 3' carbon of one nucleotide is joining. It's going to form a bond with another nucleotide, the phosphate group from a nucleotide that's at a 5' carbon below it. Okay?
So are they
Now it
still
happens again.
called? Oh,
What?
are they still called the 3' and 5' even if there's not a
Yep, they
hydroxyl
are.
group?
They're still the carbon. They still are. They still are. But you don't see the hydroxyl anymore, and the phosphate group's changed a little bit. It still has the P in it. But yeah, this is still the 3' carbon. This is still the 5' carbon of the sugar because they're designated that forever.
Okay.
But this is one nucleotide, and this is the second nucleotide. Now, pretend that this one wasn't there yet. There would be an OH there, right? Now this nucleotide is coming in. This nucleotide is coming in. It's got a base at the 1' carbon, an OH group, a hydroxyl at the 3' carbon, and at the 5' carbon, it's got a phosphate group. This phosphate is going to form a bond with the OH from the 3' carbon above. Okay? This is the 5' carbon for this nucleotide. This is the 3' carbon for the nucleotide above. Now they're bonded together. Let's say that base is a C, cytosine base. So this is a cytosine nucleotide. This we said was a guanine nucleotide, and this was an adenine nucleotide. Okay? Now I want to show you the orientation of this. At the very top, the highest point is the 5' carbon with the phosphate. So they say 5'. Okay? At the bottom, the lowest point here, is the 3' carbon with the OH. Okay? So it's when you draw it, everybody knows that there's a phosphate group here and that there's an OH here. But this is going 5 to 3 in this direction. Is everybody okay with that?
Yes.
Okay. Now, in reality, and I can't flip this up, the strands are upside down to each other. Absolutely. Absolutely they are. That's why they did these upside down, because they're called anti-parallel. They're opposite orientation. So look at this. It's kind of difficult to see this, but this is, and we're going to build it this way now. So if we look at it, this is the 5' carbon. So this nucleotide. This is the 5' carbon for this nucleotide. See, don't worry, it's upside down. This is the way they really are. This is the 5'. This would be the 3' carbon, and let's just say that that's an OH right now. And then this would be the 1' carbon. The base there, because this was cytosine here, this has to be guanine. Because C base pairs with G, and A base pairs with T. Now there are 3 hydrogen bonds between these two. C and G have 3 hydrogen bonds between them. A and T only have 2, so they're not quite as strong, but they're still strong. This is your first nucleotide over here. This is nucleotide number 4 overall, but it's the first one for this anti-parallel strand. Okay, now this is the next nucleotide. So where are the things on it? Well, this is its 5' carbon with the phosphate group, and that's going to bond to the OH group for the nucleotide at the 3' carbon above it. And you know, the H gets off. It just happens to that there's a little change because they're bonding together. The OH of one nucleotide is bonding to the phosphate group to the new nucleotide, and they form a nice bond. And now they're connected. This is the 3' carbon, and that would have had an OH on it. And then this would be the 1' carbon with the nitrogenous base. That one has to be C because I said that this over here was G. And these have 3 hydrogen bonds between them. Then we're going to go to this last nucleotide here, right here. This is the 5' carbon with the phosphate. This is the 3' carbon, and oh, there's your OH because it's done, right? Remember, this 5' phosphate is going to bond to the OH at the 3' carbon for the nucleotide that's right below it. And this is the 1' carbon with the nitrogenous base. That has to be T-thiamine, because I said that this was A, and there will be 2 hydrogen bonds between them. So when you look at this one, it's going 5'. See, this is where the phosphate is. Okay, yep, this is the 5' carbon. And here's the 3' carbon with the OH. So at the 3' carbon, you always have OH. At the 5' carbon, you always have a phosphate. And so it goes 5 to 3 this way. See how they're anti-parallel to each other. Here's the phosphate here. Here's the phosphate here. Here's the OH here. Here's the OH here. Are we good there, guys?
Yeah.
It's a little complex, but if you look at it, it's really not. And you could put in your primes if you want. It doesn't ask you to. But right here it says double-stranded. So DNA has 2 strands. And there are a lot more than just 3 nucleotides on each side. So when you look at this, if you're starting to see the ladder, if you were climbing this up, holding on to it, where you hold on become the sugars and the phosphates out here. Here's the sugars, and here are the phosphates. The steps of the ladder are the bases. So the inside are the bases, the steps of the ladder. Where you're holding on to the ladder are the sugars and the phosphates, okay? And then you twist this. That makes it a double helix. Double-stranded DNA is composed of 2 anti-parallel strands. Each DNA strand has directionality. The 2 sugar phosphate backbones run in opposite 5 to 3' directions from each other. It is important to keep this directionality in mind as you model the process of DNA replication. Okay, so let's keep going. All right, now we're going to start getting more and more into the replication, which is good. Again, you don't have models, but you don't need them. You're going to be fine. And like I said, at the end of this, I'm going to put some strands out and we're going to build the DNA. So it says DNA replication continued. DNA replication begins at specific sites called origins of replication. A eukaryotic chromosome may have hundreds or even a few thousand replication origins. Proteins that start DNA replication attach to the DNA and separate the 2 strands, creating a replication bubble. At each end of the replication bubble is a Y-shaped region where the parental strands of DNA are being unwound. The region is referred to as the replication fork. Okay, so these would have been all together. There would be no, these would have base paired with these. It would have been like a zipper. But when DNA decides to replicate, if it just had one of these, it would take it forever to do that. So you're going to get multiple of these origins of replication. And so it looks like a little bubble. But on each end of it, if you were to look at this, this is like a Y. If you go to the right, if you turn, here's one part of the Y, then this would be a continuous part. Now look at here. This is another Y here. And then this, and this, and where they're attached is another Y. And then here, and here is the Y. Do you guys see these Ys?
Yeah.
Yep, okay. Because each of these, the replication bubble, so here's one replication bubble, here's another. At ends, they have the Ys, and they're called the replication forks. This is a replication fork. This whole thing is called a replication fork. So for this one bubble, you have 2 replication forks on the ends of the bubble. On this replication bubble, you have 2 replication forks as well. So everywhere where you see this, because this is an actual molecule of DNA, you'd have a replication fork here, you'd have a replication fork here. And this one you can really see it. See the Y there? You can see it all the time, but sometimes it's sideways and sometimes it's upside down. But it's easy. So here's a whole bunch of, everywhere you see this little circle thing. See, DNA would be like this if it wasn't replicating. But when it replicates, you start seeing these bubbles called the replication bubbles. And a replication bubble has the open area, and then at the very ends of it, it has the replication forks. Now you won't be able to see this because watch your teacher create a model of DNA replication bubble using 2 mini tubers like the one shown here. You won't see that, but I think you can get the gist. Okay, it opens up. This would be the replication bubble. There's a fork here, and there's a replication fork here. So it says identify and label the replication bubble. So the whole thing is here. And here would be a replication fork. And here would be a replication fork. So it tells you, looking at the mini tuber model, what do you think might be the first step of replication? To unwind it. Start to unwind sections of the DNA because it's all wound together. Did you guys fill that in?
Yeah.
Replication bubble, replication forks. Okay, here's a replication fork, and the bubble. All right. All right. So we're going to continue with DNA replication, and nucleotides are added at the approximate rate of 50 nucleotides per second in a eukaryotic cell. The human genome contains 6.4 billion nucleotides, 3.2 billion base pairs. Now, of course, what are base pairs? That means where one nucleotide is bound to another across, like an A with a T, a C with a G. So, of course, if you have 6.4 billion nucleotides, there has to be, half of that 3.2 billion base pairs. This is counting every nucleotide. This says where they're paired, which must be copied. Calculate the length of time in days that it would take to copy the human genome. Show all calculations, including units. All right, so let's do this. Get out your cell phone, and you probably have your cell phone anyways. Let's see what we have here. See if I can find mine. Give me a second. And let's do this calculation. So they're telling you 50 nucleotides per second. All right. That's the rate. All right. So we're going to do 50 nucleotides per second, and we're going to be working with 6.4 billion nucleotides. Right. So take 6.4 billion. So that would be 6,400,000,000. Okay. That's what 6.4 nucleotides. You start with that. And then you're going to multiply it by 15. You're going to multiply it by 1 second divided by 50 nucleotides. So I'm going to set this up. 6,400,000,000 nucleotides. And you're going to multiply that by 1 second and divide it by 50 nucleotides. Okay, let me get my calculator. Hold on for a second. Here. hold on for 1 second. I gotta find it. Excuse me, I'm sorry. I had it here. Okay, so you're taking the number 6400000000 times 1 second divided by 50 nucleotides. Who has the answer for that?
128,000 days.
No, we're not going to
No.
days yet. I just want to do this 1
Oh, okay.
second. So what's 6,400,000,000 divided by 50?
128 million seconds.
Let me see, because I got something a little different. Let's see. 6,400,000,000 Oh, I know. I can't go that way. Hold on for a second. It's my calculator. That 6 times 1 divided by 50. Yep, that's right. So it's 128 million, right? 128 million seconds. Did everybody get that?
Yeah.
Okay. Now, what you're going to do is to that 128 million seconds is you are going to divide it by 60 seconds because on a conversion factor, it would be 1 minute over 60 seconds, and you'll be able to convert that to minutes. So take that 128 million, divide it by 60. And what do you get there? $128 million seconds divided by 60 seconds. You'll have minutes on top. What do you have there? Anybody get that?
2,133,333 minutes.
Yeah, that's good enough. So that comes out to 2,133,333 minutes. Okay. And then what do we have to do that? We have to divide that by 60 minutes because that will give us hours. The conversion factor is 1 hour per 60 minutes. So divide that one by 60. And what do you get there? How many hours do you get there? Anybody get that answer there?
$35,555.
Yeah, or 56 hours. You're right. We're kind of rounding. It doesn't matter. You're right. Hours. And then take that one and you're going to divide that by 24 hours. And this will give you the days. Take that, divide it by 24. What'd you get there?
1,481.
Yeah, there's something that's wrong here. Hold on for a second. We had 6 billion. Okay, I did seconds into minutes and minutes into hours. And then, yep, 1, 4, you had 1,481 or 82?
Yes.
Okay, so that's the days. All right, is that what you got to, Joe? Joe, are you still there?
Yes, I'm here. I made a mistake in the beginning.
Okay,
Yeah, when I
so
said the, yeah.
okay, so I'm gonna get off. I'm gonna go back. We'll come back to this in 1 second, but I want to show you how we did it. Here, stop sharing for 1 second. Oh no, it took. Oh shoot, I shouldn't have done that. Probably. It's like it froze. Let me put the video on. Oopsie. You're not going to be able to see this, I don't think. Let me see. It won't. Let me see.
Can find for me.
You can see it?
Yeah.
Oh, good. For me, it's telling me it isn't. So everybody could see this?
Yeah.
Yes.
Okay. You see how I calculated it, the days? I took because these are just conversion factors. They said that you know how to do conversion factors on here. So
It's a little too close.
better?
A little more. Okay. A little bit more. Okay, that still isn't the full thing.
Okay, you can
I
see it
Bring
like
it
now?
up just a
lift it
little bit.
up a little bit, just like, yeah, and then
Perfect.
a little bit closer, but lift it up a tiny bit. That's good.
Okay. Why don't you take a picture of this with your cell phone just to show how we did it? But these are just basic conversion factors. We started with how many nucleotides we would be able to replicate, right? And they gave you how many you were doing in a second. We converted the seconds to hours, to minutes, minutes to hours, and hours to day. Now that's a long, long time, isn't it? If you were just doing one replication bubble. That's why we have to have numerous replication bubbles so that we could do this in a fast time. Got it? If we only had one site of where we were replicating DNA, this would take us forever. We would die. But because we have multiple areas that are being replicated at once, remember all those replication bubbles I showed you? We reduce the time because we're not just having one site of origin to replicate the DNA. All right, let's go back.
So is this happening like all over the body all the time?
Yep, yeah. Anytime that
Okay.
you're duplicating DNA. Anytime the DNA is being replicated, that's what will be going on. Okay, so you could just put in the answer. It's approximately 1,481 days. You know, you might be 82 or something like that, or 80. Why do you think multiple replication bubbles form during the process of DNA replication? Because it would take so long if you only had one bubble. That's why you have multiple. That's why you have multiple. Okay, so then if we were to have the actual model there, because we would have had this whole thing, you would begin the process of DNA replication by feeding the strands of the constructed DNA into the helicase. Now, we're going to talk about enzymes. The first enzyme we talk about, you need to know this, is helicase. So, helicase, its job is to unwind the DNA. Now, we're not going to be able to make real good bubbles, but helicase unwinds a small section. This is the best that we could do. So your DNA, what you would have done is you would have made, opposite strands, anti-parallel strands. One would have been going 3', 5', and the other one's going 5', 3', anti-parallel. I took you through that already. And then you would have just put this on the helicase and you would have unwound a small section. So this is just one replication fork on a bubble that would have been over here. But we can't simulate exactly what happens in the body because you can't do it with the models. So we would have just unwound a little bit of the DNA. What does that mean? Open it up. Open it up. The enzyme is called helicase. Here it is right here. And what does helicase do? This is important. It unwinds the DNA. You need to know that. It doesn't unwind all of it. It just unwinds them so that you can start duplicating the DNA. And it tells you to be sure to position the 5 and the 3' ends of the DNA appropriately as you place the DNA on the mat. There would have been a big mat. Continue feeding the DNA through the enzyme until you have 11 bases emerging from the helicase. So what would have happened is you would have just been adding new bases to the stuff that you unwound. Notice that helicase moves into the replication fork, not away from it. What does helicase appear to be doing? I told you this. What does it do? What is the purpose of helicase?
To unwind DNA.
Yep, it unwinds the DNA. Helicase unwinds the DNA. Identify which type of bond is broken. They're hydrogen bonds. Remember, we're breaking the bonds between the base pairs, and those are hydrogen bonds. Bonds. Why is the helicase able to break these bonds? It's an enzyme. It's specific. Helicase is an enzyme, and remember, enzymes do specific things. It has the capability to do that. It's an enzyme. It's specific for breaking hydrogen bonds in DNA. So the first answer is it unwinds the DNA. Second answer is hydrogen bonds. And the next one is it's an enzyme specific for breaking those DNA. Specific for breaking the hydrogen bonds. Okay. Hopefully you filled that out. And we would have continued replicating the DNA. All right. And remember, the strands are anti-parallel. I think I want to take you to something different now for a short period of time. Well, maybe I should finish this first. This might be a little complex. There's only 4 more. If you don't get this, it's okay, because I'm going to take you to something else where you will understand it more. So remember when we had the DNA, we had one strand going 5, 3, and the other 3, 5? Just like this. They're anti-parallel. They're still going 5, 3, but in opposite directions. So it says, note, replication occurs on both sides of the replication fork simultaneously. For simplicity and clarification, you will stimulate the replication on one side of the fork at a time. It's occurring simultaneously, but you can't do it that way in the model because it's too confusing. So you're going to do one at a time, but it's happening simultaneously in the real world. All right, so what's going to happen? After helicase unwinds, you would have binding proteins. They're just called binding proteins. And I want you to know that too. So make note that they're just called binding proteins that will stabilize the single strands that you've unwound. So helicase comes in, it unwinds the DNA, and then the binding protein keeps them from coming back and binding to each other. And then an enzyme is going to come on, and they jumped to the end right here because they couldn't have all of this stuff. But I'm going to teach you the right way that it would have happened. But this is good enough. Eventually, what you're going to have is you're going to have an enzyme called DNA polymerase. Okay, so for the strand that was the parental strand, because you started with you started with 2 strands of DNA that were parent strands. You had one going 3-5, and this would have been 5-3 down here, okay, because they're anti-parallel. For the one, and they show you this here, okay. This is the strand that we're working with first of how we're going to duplicate. You can just start adding the complementary DNA nucleotides to it, okay? Now, there's something that happens first, but they just jumped over that because they're like, no, it's getting too complex with this model. So we're just going to add the DNA nucleotides, okay? So DNA polymerase, that's an enzyme comes on board, and here's DNA polymerase. Here's the little enzyme. It catalyzes the synthesis of new DNA by adding nucleotides to a pre-existing strand or chain. New DNA can only elongate in the 5-3 direction. So you remember when you're making new DNA for this strand here, this top one, okay? This will be easy because the strand that you're making will go 5-3. They have to be anti-parallel. So this one will be easy to make. Easy to make. You just start adding DNA nucleotides and keep going, keep going, keep going. So where there was a T here, there would have been an A here. Where there was an A here, there would have been a T here. If there was a G here, this would have been a C. And if this was a C, there would have been a G. Okay? You just add them on. You would have been bounding them. And DNA, this is DNA polymerase. This is the one that can actually add nucleotides. So it keeps going, keeps going, keep going, keep going, keeps going, keeps going, keeps going. New DNA can only elongate in the 5-3 direction. So that's okay, because it has to have an empty 3' end always. The DNA strand that is made continuously is referred to as the leading strand. So you're making the leading strand in the 5-3 direction. So simulate replication in the lagging strand. All right, so the lagging strand, because this one is oriented 5-3, and you can't just start synthesizing 5-3 of the new one because it would be just like the old one. So you've got to actually go 5-3 going from this direction going out. And this will be made discontinuously in small sections, in small sections. Okay. The one that you're going to make in small section is called the lagging strand. Let's finish the leading strand though. Okay. So again, this strand, which is 3-5, so here would be the 3' end, and this would be the 5' end over here. You could start synthesizing directly 5-3 direction, forming the leading strand, and that's continuous. Simulate replication of the leading strand by placing one DNA polymerase at the point of origin and adding nucleotides in the active site to the parental strand. Continue adding nucleotides as you move the DNA polymerase until you reach the fork. Okay. As a new nucleotide is added to the growing DNA strand, which part of the new nucleotide forms a bond with the 3' hydroxyl? It's going to be the 5' phosphate. Okay. So that would be 5' phosphate. And sketch a helicase on the diagram to the right. So helicase unwinds, so it would be right here. And it says indicate the directionality of the newly replicated leading strand of DNA. It's going, I want you to put a 5' right here and an arrow that goes like this, and a 3' down here. It didn't make it yet, but you could put a 3' right here if you want to. Note, the 3' hydroxyl is essential for adding a new nucleotide to the growing DNA strand. If this group is not present, for example, if there is a 3' H instead of a 3' OH, then DNA synthesis cannot continue. This is the basis for the development of the Sanger sequencing method, which is used in determining the sequence of nucleotides. I'm not going to quiz you anything on that at all. They shouldn't even put that in. It's too confusing. So what they're telling you is when you're adding one nucleotide to another, okay? I showed you this. The 3' hydroxyl will bind with the 5' phosphate from the nucleotide below it. Now it says right here, will you be able to synthesize the other strand of DNA in a continuous manner when using this model? No, because see, this strand, this bottom one, is going 5'3" already. So you can't start synthesizing 5'3" going this way too, because they wouldn't be anti-parallel. So what you're going to have to do is start this right here where it opens up, and this will be made discontinuously in a series of small segments. I know you learned about this in lecture. They're called Okazaki fragments. And after we get done with this, I'm going to take you through something that I think is going to be okay. Or maybe I think I might just, not sure. I want to make sure that you could see it. You'll just have to make sure when I'm drawing it that you tell me if you could see it or not. So you cannot, because this one is going in the 5'3" direction already, and you can't just start synthesizing new DNA, a 5'3" on top of a 5'3". So you'll have to go backwards in small segments. Okay, so now we're going to do the bottom strand, the lacking strand. So discontinuous replication. Place the second DNA polymerase at some point on the other strand of DNA. Notice that DNA polymerase must move away from the fork instead of toward it, meaning you're synthesizing this one in this direction, going this way, instead of the leading strand was synthesized like this, going this direction. Now the lagging is going to be going this direction in small segments. Notice that the DNA polymerase must move away from the fork instead of toward the fork, as it did in the leading strand, in order to accommodate the 5'3" synthesis of DNA. Short fragments are made on the second strand, which is referred to as the lagging strand. So this bottom strand that was oriented 5 to 3 is going to be made in small segments. Here's the first segment that's going to be made, and then you have to make another segment and another segment too. Short fragments are made on the second strand, which is referred to as the lagging strand. Continue adding nucleotides in the active site as you move the DNA polymerase away from the fork until you reach the end. So the bottom one you just make in small segments, and then you join them. So they say sketch and indicate the directionality of the fragments composing the lagging strand. So what you're going to have to do is make a bunch of little fragments. Like this one will go 5'3", and then this one will go 5'3", and then another 5'3" on them. Feed the next 11 nucleotides through the helicase and continue sliding the DNA polymerase along the leading strand, adding more nucleotides as you progress. And then you're going to do the same thing with the lagging strand. The lagging strand requires you to move the DNA polymerase. Place the DNA polymerase back at the fork junction to create the next fragment. Move the DNA polymerase so that the bases may be added from the 5 to 3 direction. You have now created a second fragment of DNA in the lagging strand. These fragments are referred to as Okazaki fragments. That's important. And are usually about 100 to 200 nucleotides long in eukaryotic cells. When your polymerase bumps into the first fragment, you will need to remove the DNA polymerase and join the two fragments together with the appropriate nucleotides. The actual process of joining the Okazaki fragments together is more complex and involves several other proteins. So they're just showing you how you're replicating the DNA. Complete the process of DNA replication with the remaining 11 nucleotides on both the leading and lagging strand. DNA replication is considered to be semi-discontinuous process. That's important too. Why is DNA replication considered to be a semi-discontinuous process? Because it's done in sections, okay? Even though you have the leading strand being continuous, it can only go so far because DNA is only going to open up so far. So you're going to do some of that, and then on the lagging, that's done in segments, and then you'll have to open up again some more and continue on the leading strand and the lagging strand. And you don't have to sketch one of these. Don't worry about that. How do the new strands compare to the original parent strand? They're anti-parallel, right? I mean, when you get done doing this, you would have 2 new DNA strands. Each half has a parent strand and a new strand. So that's called semi-conservative. And I'm sure you heard about that in lecture. Are we okay with this?
Yep.
Okay, so we're done with the pamphlet because that was something you're going to go through even though you were building. But now I'm going to take you to something that's a little bit more real world that you will understand. And it's a little bit more than that. So the first thing I'm going to do, I want to get out of this thing. I want to stop sharing now. I want to go back to my meeting. All right. Now I'm going to draw a DNA strand, okay? And I'm going to have it unwound already. So helicase unwound it, and the binding protein stabilized it. So I'm going to, and don't worry, I haven't made it yet. So give me a little bit of time. Okay, so I'm going to have an A, T, C, G. Let's see, A, C, A, T, G, A, G, C, T, A. All right. So then, and I think this will make more sense to you. Okay, so this is the first thing. Can you see it? Yes
Yeah.
or no?
Yeah, we can.
Yeah, I can see it.
Okay. All right. This is, pretend like it's together, okay? And this is only one portion of DNA that goes on and goes on and goes on, because I don't need to draw tons of it for you to see it. But do you see where there's an A on one strand? There would be a T across from it and a C and a G, okay? Do you see how one goes 5-3 and the other one goes 3-5?
Yeah.
Do you see how I put the little phosphates in the OHs so that you could see?
Yep.
Okay, so we're all good with this. This is the parental strand. Each one of these are the parent strands. All right, now, helicase comes on board and unwinds a small section at the bubbles, at the replication forks, and binding proteins stabilize it. Now watch what I'm going to do. We're going to look at the first. Do you see how the first strand goes 3'5"? Right? In this direction, right? We're going to focus on that one. When it's going 3-5 from left to right, the new one that you're going to be making is the leading, is the continuous strand. So that is not going to be the lagging one. That is the one that's going to be made continuously. That's the leading one. Okay? We're going to make the leading strand. All right, now, the one thing that the exercise didn't tell you about was DNA polymerase can't start this off. This is the truth. It needs a primer. Do you remember the primer from lecture? It's a little bit of RNA. So I'm going to underline, and you'll see this on the test where the primer will be. Okay. So do you see how I underlined 4 nucleotides? It's not always 4, but I just did that so that you could see. So I underlined 4 nucleotides. Do you see it? Everybody see that? Yes or no?
Yes.
Okay. All right. Now, what you're going to do, there's an enzyme called primase, and that's going to add RNA instead of DNA. Just a little bit, because this is really how it works. And I know you learned this in lecture. You had to. So, RNA doesn't have any thiamine. Where there would be thiamine, you put uracil in. So I'm going to put my little primer in. Here's my primer, starting at that end. Okay. Do you see the little primer I put in? Everybody see it? This is RNA. There's no thiamine. It's uracil in RNA. You got it? Are you with me here? Okay. Then DNA polymerase comes on board, and I'm making the leading strand so then I can go all the way and complete synthesis of that strand. You see how I finished it with DNA nucleotides? Using
Yeah.
DNA polymerase? You see what I did? Everybody all right? Yes or no?
Yes.
Yep.
Okay, now watch what I'm going to do. I'm going to get rid of the RNA, the primer, and I'm going to just convert it to DNA. This is really what happens, guys. Do you see I crossed out the RNA and I replaced it with the DNA nucleotides? There's no U in DNA, it's T. You see that? So when you look here at this one, UAGC, okay? That was RNA. I get rid of it. This is really what happens, guys. And you replace it with DNA, which would be TAGC. These aren't base pairing. It's replacement. It's different, right? These are all base pairing. But you're just replacing the RNA primer with DNA. Are you all right? This is really important. Okay.
So why are you replacing the
RNA?
RNA?
Because
Yeah.
you can't. It has to be all DNA.
Okay.
All right. So you got a little primer of RNA. That's the way it works. But you got to get rid of that. So now you have DNA, and then ligase is the enzyme that seals them together. It's everything together. All right. Ligase, which I'm sure you've heard of. This is called the leading strand. And you always make it, going 5 to 3, 5 to 3. Whenever you're making DNA, it's 5 to 3. Are we good there? All right, now we're going to do the bottom strand. We're going to make the lagging strand. Now this is a problem, right? Because this is over here, it's 5 to 3 to begin with, right? So you're going to have to make this in a lot of different segments. Now watch what I'm going to do. And you'll have this because you're going to be given DNA not exactly like this, but similar. And the underlined areas are where the primers are going to be. So I'm going to put primers. And in this case, I'm only going to put 2 primers, but there could be multiple ones, and I always have them underlined. So we could see that these are where the primers are going to be. That you got to, where you have the underline, that means that's where you're going to have to put the primer in. So remember, we have to synthesize 5 to 3. So this is going 5, 3, 5, 3, 5, 3. Okay? In that direction. You have to do that discontinuously because of how this one is oriented 5, 3 already. So you got to go 5, 3, 5, 3. So we're going to do this one first,
Did
and then
you
we'll do this.
did you pick those specific areas for a reason, or were those
Nope,
just like
nope,
the
nope,
specific?
nope, nope, just for any reasons I could do, okay?
Right.
All right, so now I'm going to put my primer in, and this is the first primer that would be going. Okay, here's my first primer. You see it? That's RNA. Remember, that's RNA. Okay, and then here's another primer. Here's the other primer. See it? But your body would be doing this one. Just let it go. I'm sorry, that's an emergency, but sorry. I'm so sorry about that. Okay. So are we good there? You see it? But we would be doing this one first, and then that one, okay? Then we are going to add our DNA on it, okay? So fill in the rest. A, G, C, T, A, and G, A, G, T. Okay, so now then off of the primers, I added the DNA. Everybody see that? Okay, and then I'm going to replace the primers with DNA, right? This is the lagging strand. So I get rid of my primers and put the DNA in. Okay. This is the lagging strand. And you see it with ligase. And these little pieces of the DNA are the Okazaki fragments. You should know those Okazaki fragments, the DNA that follows the RNA before you replace it. Okay? And then that's it. So you'll have to do this on a test. And that's it, guys. All right, I've recorded this. That's it. So we're done. Good?
Yeah, thank you so much.
Yep.
Yeah,
All right.
thank
Thank
you.
so much.
All right. And you can see this later. All right. Bye-bye, guys.
Bye.
Also, when is homework 7 due?
Not until a week from the Saturday. The next Saturday you see me. Okay?
Okay, gotcha.
Right. Bye.
Thank you so much. Bye.
Right.
Bye.
Hey, Jan.
Hi, Johnny.
The reason I called, I just wanted to wish you good night and thank you very much for being my sister.
Oh, you don't have to thank me for me. You did a lot for me, Johnny, throughout my life.
I don't think so. But
Oh,
thank
yes, you
very
did.
much for being there. It really means so much to me to have you there.
I know that. I know that, and I'm going to be with you forever. And yeah, you know, Annette and Tanya really like you.
I mean, they like me.
Oh, they really